Okay, let's start. Hi everybody, welcome to today's lecture on action cognition. Peter told you in the first lecture that I'll give a few of the sessions in the seminar and the lecture. Today is one of them. My name is Tim. I have others across the hall. If you have any questions, you can come anytime or not. Today we have quite a program put together with a lot of information. I'll try to fly through. If you have any questions along the way, let me know. Okay, so let's start with the usual. Let's look at a few of the questions that Peter put up last week. And let's start with the pop-out effect. Is there any volunteer, just to get you guys up to speed on how to explain it? And as usual, I'll give you a quick hint. No. So, yes?
Yeah, you can distinguish between color and orientation. So, for example, this one, A, yeah, it's about orientation. So every bar is red, except of one, so it pops out. And then there is color, so every, not every, like all the bars are red and have the same orientation, except of one, yeah, which is green.
That was a little mixed up, but the idea is...
Yeah, so like every bars have the same orientation, but different color. Yeah, so it's because one is green and yeah, therefore it pops out.
Exactly. So it's about the contrast, right? And the idea of pop-out is that if you have to search for the odd one out, and you have to tell me whether it's on the left or the right side of the visual field or the display here, then the idea is that your search times are constant. If I double the amount of distractors, it will still pop out. If I cut them in half, it will still pop out. You're very efficient in finding it, it pops out to you. If, however, you were to find some combination of features, then it takes long. It will increase linearly with the amount of distractors input. The second question I put up was about covert attention. Just if you put somebody in the lab and you let them covertly attend a certain part of space while they fixate here, what will you notice in their reaction times and accuracy on telling these letters apart? Yes?
Well, the accuracy rises and the reaction time is slower.
Exactly. And it's on cost of all the others here, so they might be worse than those. Because today we'll look at a lot at covert attention and physiological effects of covert attention. That's why I bring this up real quick, so you know there's also things you can measure in humans behaviorally that this does. Okay. Let's jump into MT. So last week you talked about area MT, which is also known as V5 in the monkey and the human. This is one hemisphere of a macaque, and you know that V1 is retinotopically organized, so things that are up in the visual field will be retinotopically mapped to the bottom, whereas the bottom will be mapped to the top. In MT, it's a little different. It's tilted a little bit. The distinction is still top and bottom, but the distinction here goes posterior to anterior. But it's still separated, and still things that are next to each other in your environment will be represented next to each other in the cortex. Trick question, this is the right visual field, where's the left? Anybody? There's just one visual field because it goes to the other hemisphere. Keep on. Good. Now the question is, MT is also known as V5, which, you know, we talked about V1, V2. I don't know. I don't think you talked about V3 and V4 yet, but they'll come up. V5, it might give you an idea of, wow, that's a high-level area. If it's V5, it's very high up, but actually it's not so much. It's just a naming convention kind of idea. It's not necessarily in order. So MT, so if you start from the retina, it has one major input from the retina to the LGN to V1, and from V1 and V2 to MT. Turns out, however, that MT doesn't only have input through striate cortex, so V1, V2, it has a little bypass route that goes through the superior colliculus, the pulvina, and then to MT. Turns out if you lesion V1, MT is still active. It's not the case that you cut away everything because it has a little bypass there. So it's not as high as you might think. Okay. The function, this is just still a recap, that's why I speed up a little bit, is if you measure neurons in MT, they're direction selective. And it's a certain direction. It's not it's not they're selective to left and right. They're only selective to leftward movement or rightward or upwards or so or something like that. And if you record things from MT, usually what you don't want to do is you don't want to show a bar that moves in a certain direction. And I think Peter said it last week why that is. Because if I show a bar and I move it, you never know, is it the movement that drives the neuron or is it the bar that drives the movement? So if you want to record something about motion, you would like to put in as little structure as you can. It turns out there's very little structure in randomly moving dots. So if you have no correlation whatsoever, all those dots go wild. They all go random. And if you increase the correlation from display to display here, more and more of these dots will move in the same direction. Turns out the more coherence there is, the more neurons will fire. And this is, I just put it in because somebody last week asked how good people are, how much of this correlation they need to pick out that things move in one way or the other. Is that person here? Hope so. So basically forget about this axis. This is just how far these dots move. Look at this. This is amount of correlation. And let's look over here. That's humans. If you take the average about it, you need about 10% correlation for people to reliably tell apart whether things move left, right, upwards, or something like that. And in a monkey, if you overtrain a monkey, they're usually better than humans in those perceptual low-level tasks. So they're at, I don't know, 5 or so. Turns out if you lesion, well, in a monkey you lesion, in the human game, of course, don't lesion, but people sometimes have strokes in that area, so you can tell. So if you lesion MT in a monkey, or you have a person with a stroke in that area, you see that this threshold goes up to about 100. So it goes all the way up. So you need a lot more correlation to have people perceive things as moving in the right direction, which might give you an idea that MT is involved in the perception of movement and discriminating movement. At the same time, if you don't lesion it, but you stimulate it, the other side of things, you have this diagram, right? Yeah? Okay. I'll go really quick. So this is this shows you that with certain correlation in some of the directions of movement of those dots, the monkey will say, well, this moves leftward or rightward or upward, downward, it doesn't really matter. And you can find a spot with 0 correlation. They should be around chance level. I don't know why it's not here. But it turns out if you stimulate MT, you can shift this curve, which means, in contrast to what is actually presented to the monkey, you can make it perceive things as moving more to one or the other direction by stimulating MT. So it has to do a lot with motion, and it might be the input to being aware of motion too. So now the question is, if we would like to look at MT in a human, how would one do it? I mean, we're pretty bad with single electrode recordings in humans, of course. So what we usually do is, if we want to localize regions, is fMRI, because it gives us quite a good spatial resolution. We can go down to 1 by 1 by 1 millimeter, depends on the field strength, of course. This is again a quick overview of how good, what the benefits and, or well, the pros and cons of the different methods are. I think you talked about most of those last week as a quick overview. You see EEGs up here. EEG can be, oh wait, no, sorry, scalp earpiece. That's what we're measuring. That's the EEG. So that goes down to 1 or 2 milliseconds temporally, but spatially it's really bad. It's like from 1 to 10 centimeters. With fMRI, fMRI is very slow, but spatially it can give you much better information down to 1 millimeter. And today we'll look at fMRI a little more. Okay, so usually Peter has a long, like few slides discussion of things. I couldn't fit it in today, so here's the 1-slide summary of fMRI. So the idea is the following. You have a subject and you have a scanner. You had a few million of dollars and you bought a scanner, now you have it. And turns out you have a 3 Tesla scanner, which is, and this is just to impress you with a high number, it's 60,000 times the Earth magnetic field in there. So you shift somebody in there, and then you show things to them. If we wanted to find MT, which is our task today, we would show them static dots and moving dots. And we would see what brain area is modulated by static versus moving. So if I show those dots to you, what is happening in the brain? So first of all, you have neuronal activity, of course. I mean, in V1 will be active, and of course, if humans have something like an MT, that will be active too. With neuronal activity, you have glucose consumption and oxygen consumption. So the amount of oxygen will go down because the neurons use it up, and then it will be signaled to the brain, we need more energy, we need more oxygen. So that is why the cerebral blood flow in this very area of activity will be increased. Turns out most of the activity is not necessarily taken by the action potentials being generated, but more about the synapses. So it's not actually true that you measure action potentials or activity that's only related to action potentials and synapses too. So now we have an increased blood flow in this very area where things are active. Turns out when the brain puts in fresh blood into the area, that's oxygenated blood. So that is blood that has a lot of oxygen. And that is why the blood oxygenation in that area actually goes up. You might think it goes down because the neurons use it up, and at first it actually does go down, but then it overshoots wildly because the brain supports much more than is actually used in that area. So now the blood oxygenation goes up in that area. Okay, we're still showing those checkerboards or moving stimuli to people. Blood oxygen goes up, and with that, the uniformity of the magnetic field of this little bit of blood there will be increased. I won't go into the physical details of why oxygenated blood has more uniform magnetic field than deoxygenated, just believe me for now. So now this all happens without any magnet being around. This all happens to you right now as I speak. So now we have the thing that we are in the scanner. And the scanner has this strong magnetic field which kind of gets all the neurons point in, sorry, not all the neurons, that was completely wrong, which makes the magnetic field, the little dipoles point into one direction, and now you put a radio wave and you push them. And you need to imagine this like, so Peter's example is always if you have a few kids on a swing, like you have 10 swings and you push them all at the same time. Okay, they will all start to swing at the same time. If they're all the same weight, same age, everything, they will go slower and be slow at the same time. And also, if you stop pushing, they will not start being desynchronized that fast. But if you have a thicker kid, a thinner kid, an old kid, whatever, and you push them all, what did I say?
It's an old kid.
Well, yeah. If you push them and you stop pushing, they will desynchronize much faster. Can you imagine that this happens? Okay, so what you can do is you push this magnetic field, and if it's more uniform, it will desynchronize slower than if it's less uniform. And that's why by this decay time, which we also know as T2, you can actually, or T2 star in that one, you can actually measure how much oxygen is in this little patch of brain that you're currently recording from. So it is a wildly implicit measure. What we want to record, remember, is neuronal activity, but what we record is the desynchronization of field uniformity that is related to the blood oxygenation, that's related to the blood flow, that's related to the glucose and oxygen metabolism that is caused by neuronal activity. So compared to EEG, where we actually measure potentials, here this is a very implicit measure, and at the same time I put that here. Till the blood oxygenation goes up, some time passes. Turns out it takes up to 10 seconds, 6 to 10 seconds, until we reach a plateau where we have a reliable estimate of how active this is. So you can guess that if your temporal resolution is 6 seconds till you get it up, this is a very slow measure. So you can't measure things that are 100 milliseconds long. That's just not possible. Okay, so now we do this. If you do this once and you look at the brain, you'll find that everything is more or less active. Of course, you're breathing, you're thinking about whatever, being in the scanner, like how long is this going to take, oh my foot hurts and whatnot. You think about all these things, but also your MT is active. And if we assume that you do all the same things while we show a static stimulus and we subtract the two signals from each other, we should find something that is more active for moving ratings than not moving ratings. Okay, so if we show things to those people, this is static zero disparity dots and moving dots. And now we take the difference between these two. You find that the area MT, which is, this is a occipital pole, it's right here. This will light up. This will remain if you subtract the two signals from static and moving. So we can find an MT in human. It's not necessarily true that this MT works exactly the same way as in the macaque, of course. That's why also it is sometimes called HMT, the human MT. Okay. Okay, so now you know how fMRI works more or less, and you know humans have an MT too. So now, given that we know about covert attention from last week and about MT single cell properties, we will look at first the macaque and see what attention does with those cells, what happens. So, the task is the following. You can have the monkey sit there and just passively fixate a dot, and then at the side you flash a stimulus. So the monkey is just fixating, it doesn't really care, it's not paying attention to this, let's say. So, you have a baseline, then the light comes on, the eye position, it has no task, it will just continue staring at the point, and you find some cell activity that corresponds to the visual stimulus, because it's in the visual field, of course. Now, if you want the monkey to saccade from this point to this visual stimulus, it will pay close attention to what happens here because it wants to be fast to actually do its task, right? So now it covertly attends there because it wants to saccade over there. So now things are a little different. You have cell activity, still more or less baseline, the light comes on, and then as soon as the light comes on, you see a modulated response. You see a much stronger response in the cell, even before the actual eye movement happens. So this is not activity that's related to the eye movement, that's activity related to the visual onset. It's just that it's modulated because the monkey pays attention to it. The same goes if the monkey has to reach over it. So it's independent of the actual task the monkey does. It can reach, it can look over. If this is a visual response here, it will be modulated, and mostly it will be up-modulated by visual attention. So if we look at this in a little more detail and we go back to MT, this is one experiment, one famous experiment that shows you what part of the stimulus response is changed by attention. So this is when the target comes on. You see here what the display was. The monkey fixates on a little blue square. And there are the little dotted things here are the receptive field of that neuron. So if something moving and moving in the right direction happens here, the cell will fire. Okay. And okay, now you have part of it that is like those dots are moving in the preferred direction of this neuron, and part of it are moving in the non-preferred direction. And now you can ask the monkey to do three things. If you can do it, you can ask it first to pay close attention to the little dot, the little fixation dot, and not attend to those things. You get a visual onset response. That is the usual. Whenever you flash something, the neuron will fire, and you get that's the transient response, and you get some tonic response afterwards. If, however, you ask the monkey to attend to the preferred stimulus, and it has to, I think in this experiment, it has to spot little speed changes in the moving dots. So now it's paying attention to this thing, which is in the receptive field of this neuron. You'll see that the transient response, nothing happens. It's the same as before. It's the tonic response that is higher here than in the neutral case. And even in the preferred case, it's even worse. The firing rate of that neuron is actually reduced a little bit compared to neutral. So the firing rate of the neuron is altered in the tonic response, not in the transient. So here, I only show you preferred and clearly non-preferred. But you know that neurons have tuning curves, right? So there's a preferred and there's a non-preferred, but there's stuff in between. Like if it moves in one direction and it moves approximately in that direction, the neuron will fire a little bit too. And now we can check this tuning curve and look at the whole tuning curve and see how that evolves over with attention or without attention. I was jumping ahead. Well, V2 does the same thing. That's the take-home message of this. Similar experiment as before with MT. You see here the transient and the tonic response. You see that it's not only an MT thing. It happens in V2 cells too. Here the selectivity is of course not moving dots. The selectivity is bars of certain orientation, which we know V2 cells are selected for. But now comes the tuning curve. So this is a cell that prefers a 0-degree direction of motion. Let's say this is 0 degree. It will respond strongly to the 0, and it will respond less, like a little less to things around it, and a much less if it's orthogonal to it. And you see here two things. First of all, there's an upregulation with attention. So the cell fires more. But you see, it's not just generally firing more. It's not that the curve is shifted up. That what attention does is it modulates existing activations. So it is modulated mostly when the cell fires most, when the cell doesn't fire much, it will not make it fire more. Okay? So it's not shifting the whole curve up. It is just modulating and increasing firing rates that are there in the cell. And the same holds for V4. There's a whole session on V4 and things like that. I won't go into detail with this. V4, part of it is color selective and orientation selective, and things work similarly there. So in many cases, the tuning curves are shifted up. There's, of course, an exception to this, which I will not cover now. I think it might be part of the seminar. Okay. Oh, yeah, there's one other thing. Also, attention doesn't make this thing be tuned more sharply. Okay? It could be that if you pay attention, that then the neuron is even more selective to its preferred direction, and this curve is then much narrower. Okay? This is not happening. So it's not sharpening it, it's not moving it up overall, it's just increasing the firing rate at the next level. Okay. Here's a cool thing though. So far I've only shown you changes in responses with respect to basic features. So this is a very neat experiment. And it might take a while to get, maybe not. So you fixate on a spot, you have a monkey fixate on a spot. And then you have a stimulus onset. There's 2 red dots, and one of the red dots is connected to the fixation spot. Okay? And the monkey has the task to saccade to the little red dot that is connected to the fixation spot. Okay? So it has to saccade to this one in this case. So now if we record from neurons that have a receptive field right here, in fact, this receptive field shouldn't care really whether this is connected or this is connected or what, because its receptive field is just right here. Okay, so now what we can do, well, this is the second stimulus. You see the input to the receptive field is here and here exactly the same. So in principle, if it was just, attention was just based on this little feature inside the receptive field, there shouldn't be any difference on whether you present this or whether you present this. Note, however, that in this case, the little thing is part of a bigger object, if you like, which has to do with the task, and here it's actually part of an object that has nothing to do with the task, because here you have to fixate to this one, and this is the relevant object. So what Rodriguez et al. Found was that there was a modulation of the neuron's response based on whether it is connected here or whether it is not connected. And this, yes?
I'm sorry, why is the receptive field in the second stimulus on the other object?
Why this one's on the left?
Yeah, I don't know.
It's just so that, I'll put it so I can show it. It just shows, it has to be here, so the visual information in the surrounding of the cell here and here is exactly the same. The only difference, so that visually the cell gets exactly the same input. If I were to shift this one over here, I would have to measure a different cell because this cell is responsive to this part of the visual field. Does that make sense? Yes. So all you change is whether this is connected or not. But this part, from this compared to this, is exactly the same from one condition to the other. So the neuron shouldn't worry about it if it was only based on the feature inside its receptor. Turns out it does matter. And this is a really cool finding because this basically shows you that attention is not only active in the receptive field. If the neuron codes for a part that is just a part of an object, it can be upregulated too. And again, you see it is in this phase of the response in this line. Okay, does that make sense? Everybody? No? Yes.
This has been measured in the MT, or not?
No. I'll have to look it up. Let's say visual cortex. I actually don't remember. I'll check that. Okay. It was more a proof of concept. Okay. Does all make sense up here? Up to here? Because I'll skip from the macaque to the human. Any questions? Ask them that. Yes?
Where is the blue curve running in the left feature? Because I can't.
Oh, I'm sorry. Just this is the difference here. So it goes up exactly the same way, and then it goes. It's lower. It's lower if it's not part of the object that you attach it. Yes.
But this is really different because it's really small. And Does this something have to do something with the
You mean the effect
connection
size?
or not?
You mean the effect size is really small? Well, it's in both cases. It's a completely preferred stimulus. Well, it's actually hard to tell. It's different area, different monkey, different lab, different everything. So it's hard to tell. To me, this seems pretty big, actually, given it's the same visual input to the receptive field, and it's just something that happens far out of the receptive field that connects this thing to something. So you wouldn't expect a huge effect because visually nothing changes, just the attention. But anyway, yeah. Okay, anybody else? Okay, so let's do the same thing with humans, and let's use fMRI to put everything together. So imagine you're in a scanner. You have to fixate a dot for a second, and you have to. I mean, fMRI scanners are not totally like a fun place to be. You're in there. You mostly have a bite bar. You have like all those pillows, and you stare at a little screen. It's like this loud magnet. So now the experimenter asks you on top of that to fixate this, and then to track those 4 dots. So everything is moving around wildly, and you have to track those 4. And then the experimenter is a mean experimenter, and he turns off the color after 2 seconds, and you still have to track them. And you have to track them for 13 seconds. And afterwards, he asks you, so which ones were you attending? Turns out this is hard. So 2 is doable. I would say 2 is doable. 4 is okay. More than 4 is really hard. So what you do here is you attend a lot to this stimulus. So what they do now is, again, we have to contrast things, because if you only look at the activation of the brain in one condition, a lot of things will be active. And you want the things that are only active or modulated in their activity by attention. So now what we do is we have people track the eyes and just stare at the same thing without tracking any. Okay? So this here and this here is basically the same condition. It's just in one case you're trying to figure out where the dots are moving, in the other one you're not. So if we tell these 2 apart now, this is the contrast. Have you guys seen a lot of those inflated brains before?
Yeah.
Yeah, good. So occipital pole, temporal lobe, frontal. Okay, right hemisphere. So you see now that in the dorsal stream, so the ventral stream goes from V1, it goes down here. It's below the brain, if you like. And the dorsal goes up here. You see that there's a lot of modulation with attention in the dorsal stream. This is area posterior IPS in D7, which I think we'll talk about later in the lecture. Not today, a later lecture. And you see, this is human MT. This is what I showed you before, approximately right from the position. It's in the sulcus here. You see that part of MT is also modulated on whether you attend or not. So you can find attentional modulation in the human MT as well, and in a lot of other areas. It's a whole attentional network, it turns out. Okay. This was a lot of information. You deserve a video. This is when attention goes wrong.
Before you turn on the video, yes. Because you put that on maximum, I actually have a feeling it's going to be very loud.
Oh, yeah?
So.
It's going to be fine. We'll see.
Right.
Be prepared.
Just because it doesn't move, that's why it's
yeah, that's a little odd, right?
Once again, it's damage to the brain, which reveals much about how the normal brain functions. The attention system is often affected after injury to the right-hand side of the brain. This leads to a condition known as neglect. Neglect leaves patients with a baffling syndrome, as if half their world has disappeared.
Neglect is a very difficult concept. It's a medical term that has been used for almost 100 years to describe a variety of behavioral conditions, that is, what patients actually do when they've had a stroke. And most of these patients have strokes involving the right hemisphere, right part of the brain. And basically what they tend to do, which is descriptive of neglect, is they ignore one side of space. So, for instance, they dress one side of their body, they write on the right-hand side of the page, they miss things on the left-hand side.
The stroke that Peggy Palmer suffered 10 years ago has left her with extreme neglect of the left-hand side of her world. Peter Halligan has spent many years testing Peggy to try and understand exactly what is going on in her brain.
Sure. Yes,
This time, Peggy, I'm going to show you this picture of a cat, and I'd like you to draw it as accurately as you can.
okay.
Neglect has very little, if anything, to do with your eyes. The vast majority of the problem arises from the brain processes involved in attention. And then your attentional system provides for where your eyes move. So in other words, if something happens in my visual field that's interesting, I'll move my eyes there. But why would you move your eyes there? Only if your attentional system indicated you needed to move there. So your eyes are slaves to your attentional system. And what's wrong with neglect is the attentional system has been damaged. You notice that this cat has got 2 tails.
No, I didn't notice that.
So you don't have it on that side, do you?
No.
Is there anything else missing out? You, for instance, look around the